The next talk we're from Professor Gemunu GUNARATNE, and he is he's the chairman of the Department of Physics in University of Houston, and he received his PhD from Cornell and conducted his postdoc at the University of Chicago, and he proposed several very interesting measurements, and included one special temporal patterns, and the parameter is referred as disorder function, and to quantify the level patterns of irregularity. And he is a member of Chicago group, which first established the anomalous scaling properties of hard tubulars. Yeah. And he also has addressed some very interesting biological problems. And today, he will talk about model-free network control as well.
Thank you very much for the invitation and the introduction. So, my talk is entitled Model-Free Network Control, and I'll start by introducing what the title means. Okay, so first of all, why do I want to worry about networks? I'm really talking about genetic networks. So why do I need to talk about networks? Let's think about the biological processes that happen inside of the cell. Yeah, so this is a schematic. The cell has a nucleus, inside of which sits the genome, two copies of the genome, one from your father and one from your mother. And pieces of this genome are read by machines, RNA synthesizers, and they produce pieces of RNA which mostly come outside of the nucleus and sits in the outside or the cytoplasm. And this RNA is in turn read by another set of machines producing proteins, and proteins do the work in the body and get degraded and are reproduced again and again and again. The problem is that like the action of these genes and proteins are, they depend on other genes. So there are interactions between different genes. And here is an example of a network associated with prostate cancer, and it's a mess. There are thousands of genes acting on each other, all of them associated or somehow related to prostate cancer, and that's what this complicated diagram is. So it's a very, very complicated system or a network. The network here has proteins and genes that talk to each other, and once again, it's a very, very complicated system. And you also know that this is complicated in diseases. Okay, and here's an example of a class of brain cancers called glioblastoma. There are several subtypes, and what I have done here is to show you the expression levels or the concentration of 30 genes outside the nucleus or inside the cells. Like I have picked 30 genes, and for two subclasses of this brain cancer, I have shown you the concentration of these genes. So red means that it's higher than normal, green means it's lower than normal. And you can see that these subtypes are, their levels, the levels of these genes in each of these two subtypes, it's very different. Yeah, but change in a coherent way. Now, it's very unlikely all 30 of these genes, in fact, there's more than 30, I'm showing you only 30. It's very unlikely that all 30 genes are defective. much more likely is that there are few genes, maybe 2 or 3 genes that are defective, and because of the interactions like this, others get, the levels of the other genes change as well. And that's what likely happens. Okay, here's another example. Like I'm talking about addiction here. Like in this example, I'm talking about addiction. The experiments are done on Drosophila, and okay, what the experimenters do is to introduce alcohol to them, and in some cases, the alcohol changes the structure of the genome in cells in the brain. These inside the nucleus, they get folded in a different way or refolded because of the alcohol. And when that happens, some animals can tolerate alcohol. Okay. And the next time you give them alcohol, they take more, like to get the effect of alcohol. So, I mean, that's one model of addiction. Here again, if you look at the gene expression levels on the outside, many gene levels have actually changed. Once again, it's very unlikely that all these genes are changed, but some of them have changed and affect the others do some kind of a network, which they haven't really shown yet. So once again, okay, what happens here then is, because of the alcohol, the genes, the genome inside here gets folded in a different way, and by having these different foldings, okay, some genes cannot be read, like, and so the levels of genes outside is different for these alcohol-addicted animals, and that changes the levels of genes in overall. So these are the levels in the animals that are not addicted versus the ones that are addicted. So there are different levels. Again, you notice that there are the levels of many genes like are different between the two groups. Okay, and once again, it's very likely that it's the coupling in the network that does that. Okay, maybe a few genes are changed because of the refolding, and the rest are changed because of the interactions here. Okay, so this explains why you would need to study networks, first of all, networks of genes to handle this problem. The next question is control. So, okay, now I have told you already that the networks are important. Okay, so the question is, if you want, for example, here, if you want to eliminate the effects of addiction, you would somehow want to change the levels of all these genes to what they were before addiction. Now, how do you do that? That's the question. So you want to have, okay, there's some undesirable state, which in this case is the addicted state. In this case, it's a cancerous state. Okay, so can you design a therapy to get it back to a target state? So a target state meaning the non-addicted state, or maybe a normal state in the tissue rather than the cancerous state. Okay, so can you design a treatment to do that? That's the question I want to ask. So it's actually to, take this network and control the whole network to a state that you know, which is a desirable state. Right, so that's what the control is. Okay, again, you don't want to be, remember I said that there was likely only a few genes that were mutated, and that made all these genes change. Okay, so to change it back, you also want to change a few genes externally, and I'll talk about how this might be done later. Okay, so that's what you need to do then. Start with this undesirable state and get to some reasonable, some target state which is inside some desirable region. Finally, okay, why model free? The problem here is this kind of complicated system. It's virtually impossible to model this. Also, the interactions between these genes are biochemical reactions. We don't know the form of these biochemical reactions typically. You can approximate them, but actually to find them is very, very difficult. I mean, even in the simplest biochemical reactions. I mean, any of these biochemical reactions have many, many constituents. I mean, it's not a single reaction, it's multiple reactions, and we don't know how to say anything about these reactions typically. Okay, so that's the problem. Then you have this very complicated system with hundreds of nodes, thousands of interactions. You want to be able to control like an undesirable state to a desirable state, and you don't have a good model to do this with. So how do we get there? That's the question. But let me just, first of all, reformulate this problem. So, I mean, if you have, I mean, think about the addicted state. Okay, if the brain cells, if inside the brain cells the genome has been folded in some way, some undesirable way, it's very difficult to get it back to the original state. Impossible, but it's a very difficult task. So same is true for the cancer cells, for example. There might be mutations. Unless you know precisely what the mutation is, you don't know, you wouldn't know how to correct it. Okay, I'm going to assume right now that the cells are in what you might call a stationary state, meaning that the gene expression levels or the concentrations of genes outside the nucleus is approximately time independent. Now, of course, it's not time independent. Any living organism, the levels of genes are going to change in time. So what do I mean by saying this? So what I mean is in some kind of an average way, it's going to be constant, but it might be moving around that average. I'm going to assume that the state of this network can be characterized by the levels of the genes in the cytoplasm. Okay. Again, it's not clear this is a good assumption. Maybe the levels of proteins might be important, maybe other biomolecules that are important. But in the end, okay, genes outside of the nucleus, like it's the genes that give rise to all of the other molecules. And so you might assume that the gene expression levels, the levels of genes or the concentrations of genes outside here will tell you something about what the state of the system is. Certainly if the system is stationary, that is going to be true. If it's not stationary, maybe it's much more complicated. The other biomolecules also might be very important. So again, I'm approximating this already by assuming that the stationary states can somehow give a sense of what the defects in the cells are. So, okay, so differences between the actions of the cells or the behavior of the cells will be associated then with differences in the concentrations of genes. Oh, by the way, the other reason why I'm talking about ignoring things like proteins is it is very difficult to measure protein levels in a cell, all of the protein levels. Single proteins you can measure, but not many proteins. Genes, on the other hand, you can get the entire levels of all the genomes by sequencing. There are several other methods, and these methods are becoming rapid. Okay, by now okay, you can measure all the levels of all the genes in a single cell. So it's in a very advanced stage. That's also why I'm using this for practical reasons. Okay, so the control problem that I talked about, it then reduces to changing the state of the system, the defective state, either the addicted state or the cancerous state, to a target state, which I know because I know what the non-addicted state is. I know what the non-cancerous state is. So I have this defective state, okay, but I know what the target state I want to get to is. Okay, so the question then is, okay, how do you move this entire, all of the levels of all the genes from the defective state to the target state? Can you do that is the question. Okay, also most importantly, okay, can you do this by changing a small number of genes, the levels of a small number of genes? Okay, so the idea then is to start with the defective state, few genes to these disease cells, and then try to control it to the desirable state. Okay, can you do this? Yeah, right now there are experiments done where the levels of several genes can be changed inside of, for example, like cancerous cells. So, yeah, so what people do, is to introduce, there are molecules that are known, liposomes that are known, that move to cancerous cells. Okay, there are chemical signatures like in cancer that this liposome recognizes and goes to that. So what people do is to carry some genes with the liposome to these defective cells. So there are targeted therapies already in use. So, if you know how to do this, at least in principle, you can do it. The experimental techniques are available. Okay, so here's an example where, I talked about cancer and addiction, but the experiment we are initially proposing is not that complicated. It's sleep deprivation in the Drosophila. Drosophila are the fruit flies. And so the experiment is this. The experiment is these animals are up in the afternoon from about 6 to about 9 o'clock. After that, they go to sleep. And they're up in the morning again. Okay, but what the experimenters do is they don't let them sleep. So what they do, they shake the container very rapidly so the animals cannot sleep. And they do this for a couple of hours. And then they take their heads and sequence them to find out what the gene, how the gene expression levels have changed. So what they know is that there's a network. So the sleep deprivation can damage them. Okay, but there's what's called a homeostatic network, which prevents damage. So somehow this network prevents damage to the animals because of sleep deprivation. However, sleep deprivation actually leads to several changes, which is true of us as well. So if you sleep deprive them and allow them to sleep, they fall asleep fast, and that's what shorter sleep latency means. If you allow them to sleep, they'll sleep a longer time. That's what that is. And to wake them up once they are asleep is harder. The sleep-deprived animals, and that's what that means. And also, interestingly, that their learning skills, if they're sleep-deprived, these animals have a hard time learning. Okay, it isn't clear these animals can learn anyway, right? Okay, but the experiment is this. You give the animals two smells. different smells, and after one smell, you shock them. After a few times of this, the animal learns, okay, not to go to that bad smell, the one that shocked them. Okay, so what the experimenters do is to send them through a maze with both these smells. They can find out how much they avoided. These guys learn very quickly. Okay, but if they're sleep deprived, they have a hard time learning. So okay, these are behavioral changes in this animal, quantifiable behavioral changes. And so also the genetics, like I said, what they do is, so okay, first take a non-sleep deprived animal, so okay, take the cells in their heads, sequence them and get, 20,000 values of genes. Then they sleep deprive them for 2 hours, okay, get their head cells, sequence them, and once again get the levels of the genes. Do it for 4 hours and 6 hours. Okay, you don't want to sleep deprive them for more than 8 hours because they die. Okay, so it's 0, 2, 4, and 6. And then they ask which genes, the levels of which genes systematically increased or decreased. And they identify like some 159 genes in the head whose levels have changed. So very likely these 159 genes are in this homeostatic network that reduces damage due to sleep deprivation. However, so Drosophila is the animal whose genetic interaction is best known, like among all animals, and only about 50 of these 159 are known to be interacting with each other. So we have a long way to go to understand even this network, this sleep deprivation network in Drosophila. However, we do know what genes are likely involved. And okay, what I've shown in this network, there are about 50 of these genes which are known to have interactions within that group. So to complete this check, okay, will take a long time more. Okay, the goal again, in this case, is to take some of these genes. So okay, can you change a few of these genes to either go from sleep deprived to a non-deprived state or vice versa? And then, of course, in this experiment, we can check if these quantifiable behavioral changes are seen in the genetically modified animals as well. Okay, so that's what the experiment, the proposed experiments. We haven't done this yet. We're trying to get funding for that. Most importantly, this should be done by externally changing only a small number of genes. Otherwise, you cannot change a large number of genes reasonably. Okay, so let me summarize what I have told you so far. Any biological process has that there are a number of genes associated with that biological process and these nodes, and there are interactions between the nodes. These things are not really known. And I'm assuming that the state of a system that I'm going to change can be specified by the node values or the gene expression levels. And again, that it's an assumption right now. Okay, so the proposal is this. One thing you can find is I can take a number of genes. Maybe I can change one of them. I can increase the levels of one or a few of them. I can see how the system responds. Okay, system response meaning I can find out how the rest of the network, the rest of the genes, how their levels change if I change one of them, any one of them. Okay, I add this gene that I have picked. Okay, and I introduce that into the animal. okay, let the animal, let the level settle down. I do a sequencing experiments later, and then I find out how all other genes were affected. So I change one of these genes, and I check how the system responds to it. And okay, that can be done easily. Biologists do this. Again, the idea, what I'm going to show to you is that these responses, as I change a few of these things, I can find out how the system responds to, if you knew the response surface, how the collection of these responses to changes in a few gene levels, okay, from that, I can figure out how to calculate, how to change the system, how to implement the control. So all I know, all I have to know, is what these response surfaces are. I don't need to know anything about the network, but I need to know how the system responds to perturbations, and that's enough to design a control method, and that's what I'm going to show you. Okay, one of the things that simplifies the problem somewhat is these response surfaces are typically smooth surfaces. Not always, but typically smooth surfaces. Okay, so you can approximate them by planes or lines, and so on. It's an approximation, and this approximation can be got by calculating the responses to a few changes, and that's what gives, so allows you to do a small number of experiments and figure out how to control the system the way you want it. Okay, so let's see how this works. Okay, suppose I have a network like this, and I'm changing one of the nodes. So I want to see how the system responds to changes in that one. So I either increase that or decrease the levels of that one gene, and I can find out how the others change. Again, I can do these experiments. People do these experiments already. So X1 is the concentration of that one gene, or the average concentration, and I change the concentrations. I can find out how the rest of the genes behave. So this vertical axis here, is the concentrations of all. It's a high dimensional system, but I'm showing is just one of the nodes. Okay, so okay. Okay, so now, like I said, this is going to be a smooth change. So as I change this, generally, most of the other the levels of the other genes would change slowly as well. Evolution would have taken care of it to make sure it doesn't change very much. If one of these genes change and the others change a lot, the animal is not going to survive. So very likely, evolution would have corrected for this already. So what I'm saying is that this is a smooth curve then. Okay, if it's a smooth curve, I can take, say, 2 points and get a reasonable approximation. If it's not good, I could get another point, experimentally get another point, meaning I change the levels of this, find out how the rest change, and I can get a better approximation. Turns out I don't even need a better approximation than this linear approximation. Again, I'll come back to tell you why. So I'll use a low order approximation. So in this case, I'm taking a linear approximation, this using 2 points. Okay, now suppose my target set, the one where I want to get to, if it turns out to be near this line, okay, then I'm done. All I have to do is to take this one gene, okay, externally add or subtract so that its value is at here. Okay, the system interactions then will carry the system there. Again, that's close enough for my purpose. So I have Okay, if this target turned out to be close to here, I'm done. Okay, I have found one gene which I arbitrarily picked, and then all I have to do is to make sure that gene is at this value. Everything else will sort in. The interactions between the genes will take care of the rest of it. Okay, but of course, that's a very high dimensional space. Okay, so this is very unlikely to happen. I have a target state. I randomly pick the initial conditions. Okay, it's very unlikely that this line will be close to the target I want to get to. So what's more likely to happen, which I illustrate like this, is my target is nowhere close to this line that I have. Okay, which is to say, okay, that changing, so that gene alone, the one that I randomly picked, is not enough to get me to near this target state. Okay, that's the more likely scenario. Okay, so I need at least one, like I need to be able to control at least one other, okay, one or more genes in here, like to be able to get close to the target. So okay, the question is, out of all of this, okay, how do I pick the next one I want to be changing to get this control? Again, remember the point is to get close to this target state which I have defined already, the cancer-free state or the non-sleep deprived state in Drosophila, or like the non-addicted state. So that's where I want to get to. Okay, yeah, so the idea is very simple. Again, we use geometry for this. So okay, when I say that this point is far from here, what do I mean? There's some Euclidean distance, maybe a weighted Euclidean distance that that gives the distance, and what happens is that distance, the best I can get to, is not small enough for my purpose. So let's see what happens. Let's take a extreme case. So okay, let's say this difference is only because of one of these genes. Okay, so everything else, so just by changing this x1, I can, so okay, suppose I can change everything else close to here, but this one gene cannot be brought close to the target. Maybe, so okay, if that was the case, if in the extreme case that, okay, this distance is large because of a single node, okay, how do I find out what that node is? Just from the data I have. Okay, the trick is very simple. Okay, if the weight for that node was 0, okay, I would get to this point close to that, right? So if I redefine my length measurement so that the W, the weight of that for that node was 0, I would be able to get close to that. So in the extreme case that there was this one gene which was making the problem, all I needed to have done was to set the weight of that gene to 0 in this distance measurement, and I would know that I can bring everything else close to the target state. So, okay, so the trick then is, oops. So okay, the trick then is to set one W at a time to 0 and find out what the value of delta is. Look for the smallest value of delta with these redefinitions. So I'm ignoring one node at a time and seeing what the delta is. When that delta is minimum, so okay, take the corresponding node as the one that I should be changing. Okay, so it's a very simple idea. All of it is got from data. All I do is to calculate, redefine the expression of delta by setting one of these weights to 0 at a time, and then figuring out what the smallest value of the delta is. That then gives me the second node that I want to use for my control. So, okay, now what happens is I have two nodes that I'm changing externally. Okay, so I have a two-dimensional a set of values that I'm controlling. Again, I have the values for every other node. It's going to be a surface now rather than a line. Okay, now what I have to find do is to approximate this surface, and the approximation can be got by a planar approximation, which requires one additional condition. Remember, I already have done 2 experiments to find 2 points. Now I need 1 more point, and I can approximate the surface by a plane. Okay, now the question is my target going to be close to the plane? If it is, I'm done. I could get to the target state by controlling these 2 genes. If not, okay, I got to go through and do this again. So I have to add another gene to my collection that I'm changing. Okay, the interesting point, is when the network is connected, okay, I don't need too many genes to get close. So when delta equals 0 is the place where you want to get to. You want the target to be very, very close to here. So typically with a small number of changes, okay, you get very close to the a target state. It's not 0 because of the nonlinearity. Yeah, right. Remember, I'm assuming that this approximation is linear. Yeah, but otherwise, it converges very quickly. With a small number of genes, you can get close to the point. One thing you need to remember is I have this large node. So maybe the defects happen because of 3 nodes like that, 2 or 3 nodes like that. Okay, some 3 nodes. I don't know what these nodes are. It doesn't matter. If I start from any node, okay, and go through this process, I'll get different set of nodes which I'm correcting, but because of the connectivity, I can get close to that anyway, close to the target state. Okay, where all the genes, the levels of all of the genes are close to where I want them to be. And I don't have a proof of this, but it works in many model systems. Also, we also tested this in an experiment. Anyway, just the outline again. Again, the idea is to use response surfaces for control. Okay, systematically increase the number of control nodes, and only a small number is needed to get close to the target. Again, the lack of, this doesn't come to 0 because of the nonlinearity. So that's what I've said so far. So like I said, we have tested this on many model systems, also an experiment. The experiment is it's a nonlinear. We've done this on several electrical circuits, but the nonlinear electrical circuits are made, they're constructed using transistors. Okay, see the squares are transistors. The network itself is made on a breadboard, and we have these transistors and also resistors in this network. It was constructed to make a network approximately like that. There are 4 levels of top level nodes controlling. So 3 nodes in the next level and several nodes at a next level. So there's 2 levels of control. That's what we built it to be. Each of these the IV characteristic, the current versus potential of these transistors. There were 4 different transistors that we used, and they were like that. There's no analytical formula for that anyway, and we didn't use it. Like to get inhibition, you could have a construct another circuit. Now this circuit was not linear but close to linear, but that's the only one that we could make. So anyway, basically what we did then was to create this nonlinear electrical circuit, and then the node potentials in this system could be controlled by using a little board. And so what we did was to control this at 16 nodes, and since then we have gone up to about 40 nodes. So we control a few of these nodes and measure the rest. That's what the experiment is, basically. And yeah, so the analog of the gene expression is the node potentials, and the analog of the interactions is the current between 2 nodes. So that's what the analogs are. And here we did this, all the calculations. The mean magnitude of the potential is about 5.2 volts. I'm giving you that number to give the error estimates later. So like we randomly picked up node potentials that we wanted, and we could always get to those node potentials by changing a few the levels of a few nodes. So that's what the result is.
These circuits, are they guaranteed to have a unique, stable, steady state?
No, they're not because of the nonlinearity, but we almost always found them to be the case. Okay, now here we don't have the luxury of evolution, but sometimes we would have runaway results for this. Yeah, but very rarely that we didn't had multiple solutions, or we found multiple solutions. Actually we never did. I mean, when they converge to a single value. It's of course hard to say no, because you cannot. I mean, we didn't know how to. I mean, we had to change all 16 nodes to get a different one, and so yeah, so there could be. We didn't find any case though. Now, so one thing I want to point out is, I could improve the approximation using a quadratic fit rather than a linear fit. I mean, what I did was to approximate that the response surface by like a linear or planar approximation. I could do a much better approximation by taking second order quadratic approximations. But what happens is the following. So without noise, okay, the planar approximation at the corrections in these regions are 152 millivolts compared with 5 volts. And if you do the quadratic approximation, it's a factor of 6 better in this case. Okay, however, if you add noise to this data, 5% of noise to data, and the planar and the quadratic approximation are pretty close. And if you have 10% noise, the correction doesn't make sense. And okay, one thing that happens with genomic data is that there's variability at least at a level of 10%. And I'll talk a little bit about that later. But okay, so there's really no point doing the extra experiments to get a better approximation to this surface. Okay, okay, one more point I want to make. This is what's called epistasis. Epistasis is a word that's used by, many biologists use it. Okay, sometimes not all with the same meaning. Yeah, but the idea is to see if like linear approximations are good enough for gene expression levels. So roughly, now I'm translating this a little bit, but roughly the idea is you change the level of one gene, and okay, look how the cell or network responds. Again, how do you measure it? It might be like the survival rate or the levels of other genes or whatever. Okay, then they change the second gene. Again, ask how the survival, like in a bacterial colony, for example, or the survival changes. So you change gene A, find out how the, like some phenotype changed. Okay, now do a change in a second gene, ask how the phenotype changes. Okay, and the statement is, if you change both these genes, you should get the sum change of the two, to get the two previous cases. Okay, so like in other words, okay, you change one gene, find out how much this change the second gene, find out how much the value changed, and then add them. Okay, you should get the sum of the changes. Okay, if you try this on the gene networks or on the electronical circuit, this fails. Okay, the problem is this. Actually, what happens is this is a network. Okay, so if I change one gene, the other is going to be affected as well. So it won't be like that, but it'll be something like that. That because of the network connections, if I change one gene, others are going to change as well. Okay, same thing with the second gene. If I change that level externally, this first gene would change as well. Okay, so it would be something like that. Again, if you change both of them, okay, it should be the sum of these two things. Again, once you do that, the approximations for the model systems and for the electrical circuits work very well. And so you got to be careful about this test for linearity because it's a network. Okay, remind you again what I have done. I've used response surfaces to control, and I've told you how to control the network. Start with one node. If it is not enough, okay, find out what the next best node to control is and go on doing that. And the convergence happens, you can get it by controlling a few genes. And the lack of convergence is it's because of the nonlinearity, but it's not worth correcting for the nonlinearity because of the noise in the data itself. Okay, so this thing, we haven't done these experiments yet. Okay, so the question is, are there other predictions that can be tested? I mean, are these approximations, looking at the response surface and approximate the response surface with a linear approximation, okay, how good is it? Are there other testable predictions? So okay, it turns out like there's a very simple one. Okay, so if I change, so get two genes like this and construct the approximated surface, if I change both these things, if I make a change both these simultaneously, I get to another point, and that can be found from the planar approximation. Okay, so now that's an easy experiment to do. You can do single knockouts of animal cells. I mean, you can do two single knockouts and then test the double knockout like the levels of these genes. First of all, so this experiment was done by one group on E. coli. They looked at the oxygen deprivation network of E. coli. This is Covert et al. Back in 2004. They did the experiment. They looked at 5 what are called transcription factors that are, those are proteins that act on a lot of genes. So there were 5 of them that they studied, and they studied one double knockout. And they had the gene expression levels for all the genes. like in these 6 mutants. Now, okay, we went to the gene, okay, what are called gene ontology tables, which tell you how, what are the genes in the network, and we found about 300 genes that were in this oxygen deprivation network. And okay, so we did the calculation, looked at the single knockout, and then predicted what is double knockout, and checked the prediction with experimental data. So this was we didn't put in any known thing about the network. The only thing that we got from the network, okay, was these 284 genes that came from the gene ontology. Turned out that 70% of the predictions were within the 95% confidence levels. Okay, without putting, the only thing we did was this idea of the response surface, and approximating it linearly. And okay, nobody has ever been able to predict anything at this level with very sophisticated work. Okay, some of the things that were wrong. Okay, so one of these genes, I don't remember which one. It's PHOB. This gene, this gene was one that was right, and here were the predictions using a bootstrap method, and the actual experimental results for the double knockout were like this. Okay, so this is what happened in 70% of the cases. Okay, occasionally, okay, so something called CRAB is an example here. The predictions were like this, okay, and the results of the experiment were like that. They were completely wrong. They were outside of that 95% confidence interval. Okay, I'm choosing this because if you look at this system, this gene, the gene and the protein activate each other. Okay, so it's immediately a bistable switch. Okay, and bistability means that you might run into the other branch from this branch. Okay, so this is a very simple example where we immediately saw bistability. So here's an example of a question that you asked before. And the other examples are, we've not been able to find such a simple example among the other ones, but it could be something like bistability in these networks. We don't have the experimental results to check that. So okay, once again, the proposal, I'll very quickly go through that, is to get these sleep-deprived animals, and so already we know that there's a regulation, upregulation of 114 genes and downregulations of 55. So we know the initial state, which is the normal state, the non-sleep-deprived state, and we know what the sleep-deprived state is. So the idea is, can we genetically modify them to get there? And that's what the experiment is. And here are some of the genes that we can work with. This marked-in gene. These are the highly connected genes or transcription factors here. It is to be done. So okay, meanwhile, again, we could do that double knockout prediction. Remember, we could, from the single knockout experiment, we could predict the double knockout. And okay, so this we did. So each of the single knockouts, my colleague actually goes through 10 generations of animals and then look at, so they start with a single knockout, and 10 generations later they do the experiments to find out what the expression levels are. And so, okay, in this one case that we studied, we made 45 predictions. The good news is all of them came out within the 95% confidence interval, sorry. That should be 95%. The bad news is the error bars are a little too large, and we'd like to get a better, smaller error bars. But anyway, with the data we have, the predictions were within, all of them were within the 95% interval. So control experiments we haven't done yet. The same thing can be carried on to periodically driven networks. like for example, circadian networks. In this case, we have only modeled the results. So it turns out that so you have a network of elements which are driven periodically by some okay, it's called I mean, externally it might be driven by light or heat, but there's one cell in the body that drives these periodic networks. And the only difference, so it turns out, rather than real numbers, you have to give complex numbers in this model. In these models, complex numbers because they are the phase and amplitude both associated with the system. Okay, so here's an example of circadian behavior. Okay, but we could do the predictions again. The only difference is that these response surfaces are in complex space rather than real space. Okay, so again, the predictions were really good. We could drive it to any place where we wanted to. Okay, so let me summarize the results. What I have shown is, okay, so these biological networks associated with, with some biological process involves a complicated network. I mean, again, I'm using the network from the cancer network from earlier. In the model-free approach, you don't care about the network. All you care about are the response surfaces or approximations to response surfaces, and you get that from experiment. And okay, this is somewhat similar to looking at a landscape. Okay, so this picture is from Peter Paul Rubens. It's a beautiful painting. There's every leaf and every branch that is that's way back here gives all the details of this landscape. Or you could think of like a Chinese landscape, okay, which is an abstraction. Right, so this is like doing that. I mean, trying to give a model of realistic model of the gene network. This is more like looking at something like only a little bit that you want to see. Okay, what I hope is that I have convinced you that in some cases this is that might be a better approach because these are complicated problems, and to go through all of these details might be too hard. So the model-free control, again, it only requires the response surfaces, these blue surfaces, and low-order approximations, so it seem to work well. Again, might have something to do with the evolution. Okay, generalizers, I didn't go through the details here, but generalized circadians or any periodically driven networks, all that happens is that the surfaces are in complex space rather than real space now. And so proposed applications to, the proposal that we have made is to sleep deprivation and addiction, which I have talked about before. Okay, and the last thing is anxiety. Okay, how do you know that these animals are anxious? Okay, biologists do magic with it. What they do is to release the animals into a closed arena, like a small circular domain. And what happens is, if it's a new arena, they actually are very anxious. They go through and look for the details of the arena. But once they have gone through a few times, okay, they slow down and in fact stop at the end because they are comfortable with it. Okay, but there are mutant animals, okay, who don't stop. Okay, they keep going around. These are the anxious animals. It's a model of anxiety. Okay, so again, the question, like is one of our proposals, is to see if this anxiety can be eliminated by modifying a few genes. Okay, so these are, unfortunately, this stuff is in proposed state. I have shown you that predictions is from the response surface approach. Actually, there are predictions, okay, which we have tested and works out well. Thank you very much for your attention. And I'll provide some publications.
We can take some questions. Any questions? Okay. Yes.
So is this kind of model like a feedback loop system?
All of it is, yeah, right, the feedbacks are important, right? Remember that, I mean, 30 or 50 genes would change, but the idea is by changing a handful of genes, you can then make all of them change back to where they were. Remember so you have the normal state, and the defective state, I mean, like I told you, very likely came because of 2 or 3 changes, I mean, 2 or 3 mutations maybe. Like in a cancer, you cannot have 50 mutations, or just 50 independent mutations. It's very unlikely, right? Way before that, there'll be cancer. So, yeah, so the idea is, can you move it back by changing, also changing 2 or 3 genes, and that's what seems to be happening.
So it's using minimum changes, right?
Right, some kind of minimum changes. Unfortunately, you don't know what the original genes that change. But if you look at cancer, for example, you have this red and green things, and there's 50 genes. You have no idea what was the cause of the original change. So instead of that, you need to start from some random initial to notice this. The nice thing is, if the mutation happened because of changes in A, B, and C, you could change D, E, and F and still get to that state.
So another thing is how mature this kind of model is, because it seems that it's just like an early stage of prediction.
But
How mature is this kind of model for approximation or predictions?
So in the model systems and the electronic circuit, electrical circuit, it works very well. In animals, you have to do the experiments to get that, and in the experiment is impossible to make any prediction. So that the things that there's another complication that comes about, because even in cells in one tissue, so if you look at my tissue and look at my liver tissue, there's a large cell-to-cell variability. If you take one cell and a nearby cell, they have different expression levels. Okay, however, it turns out that these expression levels fall into a few groups. So if you do a principal components analysis, the first 2 or 3 principal components will tell you the entire variability. So that probably has to be brought into these studies as well. So it's even more complicated than what I kind of outlined. Okay, but there are simpler ways to characterize it anyway. So in this case, using principal components, and there's something called attributes, which people use also to classify these different cells. Okay.
Any other questions? If no questions, let's take